Hello learners, myself Dr. Poonam Patil. Today I am going to discuss about Indian Senna. So this Senna drug is from the glycoside, that is anthraquinone glycoside, and it is well-known drug for the glycoside. So synonym name for this drug: Senna leaf, Senna folium, Senna ki patti, Tinevelli Senna, Cassia Senna. Biological source: it consists of dried leaflets of Cassia angustifolia, belongs to family Leguminaceae, and it contains not less than 2% anthracene derivative, which is calculated as in the form of calcium cynoside. Geographical source for this drug: it is cultivated and collected in India. So cultivation mainly done in Tinnevelli, Madurai and Ramnapuram district of Tamil Nadu. Cultivation is attempted in Kudappa district of Andhra Pradesh, and it is also cultivated in Kutch in Gujarat state. Then this drug also have some historical background. So Senna was known to physicians from very old days and was included in Unani medicine. About 26 species of genus Cassia genus have been reported to contain anthracene derivative, either in free form or in the form of salt. Out of them, Cassia angustifolia, that is Indian Senna, and Alexandrian Senna, that is Cassia acutifolia, are official in different pharmacopoeias because of their cathartic activity and also because of they are available in large quantities. The other species also have some cathartic activity like Cassia alata, fistula, ovata. Then next is cultivation collection. For the cultivation purpose in India, around 25,000 hectares of land under the cultivation, which producing about 22,500 tons of leaves and 7,500 tons of fruit per annum. So Senna mainly cultivated in Tinnevelli, Ramnathpuram district and their adjacent areas in Tamil Nadu state, and is exported by ships from Tuticorin port. So Senna is a small herbs which having height about 1.5 meter. Then it bears the compound pinnate leaves. Each leaf has got 3 to 5, 7 pairs of leaflet. For the cultivation purpose, they generally require red loamy, coarse, gravelly soil, and it grows alluvial loamy soil. Before sowing, the land is plucked twice, harrowed, and leveled. First sowing is done in the month of February to March, while second sowing is carried out in the October-November after rains. Normally, sowing is done by the broadcast seedling method. For the earlier germination, seed surface is saturated with sand. About 15 to 25 kg seeds are required per hectare of land, and seeds are grown thinly. The plants are grown as semi-irrigated or a light irrigated crop. Heavy irrigation waterlogging is strictly avoided. Also, plant cannot survive very low temperature. The plants are allowed to grow for about 5 months till the growth of flower stalks occur. Though it is leguminous plant, still the application of nitrogenous fertilizers is required. After 2-3 months of sowing, the harvesting of leaves is done in 3 stages. First plucking is done when the leaflets are thick, full grown and greenish in color. After 1 month of first plucking, second plucking is done and last plucking is carried out after the 4 to 6 weeks, and normally at that time the plant is uprooted after the third plucking. So it is observed that the content of senoside in leaves is maximum when they are fully grown. Senna crop which is taken after rice crop on same land in same season, which gives the best yields, and senna grows on dry land without proper irrigation gives poor result. After collection, leaflets are dried under shade or indoor by spreading them in a thin layer. Drying takes place within 7 to 10 days, and leaves show the yellowish green in color. When it turns to yellowish green in color, at that time it is separated or it is carded, and then it is packed in a gunny bags under hydraulic pressure. So during storage, drug should be protected from light, but generally it does not lose the potency at least up to 5 years. About 1200 kg of leaves and pods are obtained in 1 hectare of irrigated land, and 700 100 kg of drugs from rain-fed land. So this is about cultivation and collection of Senna leaves. Next is macroscopic character. So Senna generally bears yellowish green color, slight odor, taste is mucilaginous, bitter and characteristic. Size of these leaves is about 7 to 8 mm in width and 25 to 60 mm in length. And shape of these leaves are lanceolate. Margin is entire. Apex is acute with spine at top. Base mostly asymmetrical at the base. Transverse lines are present, which is more prominent on lower surface. Then microscopical features. So these leaves generally having isobilaterally in nature because the palisade tissue are present on both the side below the upper epidermis and upper the lower epidermis. So nature of these leaves is isobilateral leaf. Then epidermis shows the presence of unicellular conical thick walled watery trichomes. So these trichomes are present on both the side. Then it also contain ground tissue, that is parenchyma, sclerenchyma, and this is the collenchyma. In vascular region, there is the presence of crystal sheet pericyclic fibers which are lignified in nature. Xylems also lignified in nature. Phloems are non-lignified in nature. Then it contains parasitic stomata in epidermis cells. Then spongy mesophyll and conducting tissue are presented by the leaves, which is shown in figure. The patches of sclerenchyma towards upper epidermis and above the xylem is present. Then next is the standards according to pharmacopoeia. So when Islate number for the Indian Senna, that is 19.5 to 22.5, and for Alexandrian Senna 25 to 29.5. Stomatal index 17 to 20, and for Alexandrian 11.4 to 13.3. So this is about the standard ratio according to pharmacopoeia. Next is chemical constraint. So this Senna drug generally contain anthraquinone glycoside. The most prominent glycoside, that is Senoside A and Senoside B, which is not less than 2.5%, and which generally exhibit the purgative property. Teuton scientists, Teuton in the year of 1930 isolated rehin and aloimidine from same drug. Stoll and his colleagues in 1941 reported isolation of crystalline Senoside A and crystalline Senoside B from this Senna drug. Senoside A and B, these are the stereoisomers of each other. They are dimeric glycoside with rehin dianthrone is a glycone molecule, and chemically it is known as 10-10-bis 9,10 dihydro 1,8 dihydroxy 9 oxanthocene 3 carboxylic acid. So this is the chemical nomenclature of this molecule. In Senoside B, the glycoside is in mesoform, whereas in A it is dextrorotatory form. So this is the difference between this A and B Senoside. So this is the group according to 10-10 dash, and this is R. Then purgative activity of Senoside A and B is about 40 to 60%. It is reported that sugar part of this glycoside has transporting function for the glycone up to large intestine, and it also protection action, so that oxidation of glycone to other very less active anthraquinones is prevented. Then Senalip also contain other anthraquinone glycosides in small amount like Senoside C and D, rain, camphorol, aloimodine. It contains phytosterols, mucilage, resin, salicylic acid, chrysopanic, and calcium oxalate. And it is reported that besides anthraquinone glycoside, it also contains two naphthalene glycoside, that is dinevaline glycoside and 6-hydroxymucin glycoside. For the extraction purpose, TAS auto method generally preferred, and here Senoside is converted into calcium Senoside and then it is stored. Then chemical test for this anthraquinone glycoside, that is Born-Traeger test, is employed for the purpose of detection. So drug is boiled with dilute sulfuric acid, then filtered. Then to the filtrate add benzene or other organic solvents. Then shake well. Then separate the organic layer. Then add ammonia into the organic layer slowly. Ammonical layer shows pink to red due to the presence of anthraquinone glycoside. So this is the main identification test for this Senoside glycoside. Then assay. So Senna is assayed either by chemical or biological method. For the chemical assay, TLC, spectrophotometric or the spectrocolorimetric method are employed. Then uses. So senna and its preparation are used as a purgative in habitual constipation. Anthraquinone Anthraquinone glycosides of senna are absorbed first in intestinal tract, after which the glycone part is separated and excreted in colon. And these excreted anthraquinones irritate and stimulate the colon, thereby movements are increased due to local action. And which increase the peristalsis also cause the reduction in water absorption, and it ultimately forms the soft and bulky faces. So this is about a mechanism of action. So it is believed that gripping effect caused by senna is due to the resin, that is imodin. Then adulterants for this drug. So Tirunelveli senna is adulterated with dox senna, which is known as Cassia ovata, Palte senna, that is Cassia auriculata, and Mumbai mukka and Arabian senna. So dog senna leaves are ovate in shape with tapering apex, and they show the papillose cells in epidermis, which contain 1% anthraquinone glycoside. Next adulterant, that is Paltesina, which is characterized by absence of anthraquinone glycoside. The leaflet shows the long hairs when the leaves are boiled and with chloral hydrate solution. And third one, that is leaflets of Bombay, Mica, and Arabian Senna, which are brownish green in color, more elongated. So this is about Indian Senna. Thank you.